Evolution can be kind of artificially divided into two categories: microevolutionary changes and macroevolutionary changes. Microevolutionary changes, by and large, are kind of what we've been talking about so far. They're little tweaks to the allele frequencies, how common certain genes or alleles are in a population over time, as a result of some evolutionary event. Microevolution just changes how common a certain trait is in a population, or how common a certain gene is in that population from one generation to the next. Now, there can actually be several different causes of microevolutionary change, of which natural selection is just one. What other kinds of processes could drive a microevolutionary change? Well, mutation is one that we've already talked about. Through mutation, a gene that did not exist before is going to suddenly be in the gene pool. And if microevolution is just a change in how common certain genes are, and you have something brand new, then definitionally that is a microevolutionary change. And this gives us new kinds of traits. It gives natural selection something to work with. But not every mutation is going to lead to adaptation. Mutation just gives us variety. It gives us variation of traits. Natural selection is what kind of screens the traits out and decides which ones are advantageous, fit, or non-advantageous, unfit, the ones that help reproduce or do not help reproduce. There are other possibilities as well. There's something called genetic drift. Not every single mating of two organisms is going to be driven by a strong natural selection pressure. Sometimes genes become more common in a population or less common in a population entirely by random chance, and we call that genetic drift. Now, genetic drift really only happens with small populations. You don't have to worry about it too much if you have a really, really big population. Those kinds of tiny little statistical fluctuations average out. Then we also have something called gene flow. Gene flow is related to how organisms move around. Let's say that we have some ancestral organism 1,000 years ago, and they have 5 kids. 3 of them die, but 2 of them go off and start their own family lines on different islands. So they've been separated. Now, the organisms on island A, generation by generation, they're going to get new mutations that don't show up on island B. They're going to go through their own evolutionary process. They'll have a distribution of traits on their island which is somewhat unique to them. Over here on island B, they'll have their own mutations that are different. They'll have their own evolutionary processes, their own selection pressure. So these 2 islands will have different populations, and you'll be able to tell which island someone came from by sequencing their genomes. But then let's say that an intrepid explorer from island A decides to go over to visit island B. Well, they would be bringing all of that genetic diversity, all of those new genes that have developed over the past 1,000 years, over to island B. And this is what we call gene flow, when individuals from one breeding population migrate over to another breeding population. That dramatically changes the distribution of genes inside island B, because there's all this new genetic diversity coming in and being delivered on that organism. So that's gene flow. So let's go through each one of these one by one, look at some examples. Ultimately, all genes arise through mutation. Every kind of gene is different from all the other genes because at some point there was a mutation in that sequence. Here we have 2 kinds of moths. Now they are the same species. These are the exact same species, but we have 2 distinct phenotypes. That moth with the white wings with a little peppered appearance, that was the most common phenotype of this species for a very long time. At some point in history, a little mutation happened in one of the members of the species, and you got this one on the right, this all-black colored moth. The peppered moth lives in beech forests found around London, England. And if they land on those beech trees, then, well, they blend in pretty well, because beech trees have this nice white bark with the lenticels, these little kind of dashes on them. So having this white coloration with these little black dots, it's actually pretty good camouflage. And they don't want to be eaten by birds, so they avoid them by kind of blending into the bark of the tree. This black-colored moth, the dark-winged variety here, this mutant, he's not so well adapted to that condition. He's going to stand out like a sore thumb if he lands on that nice white bark. So most likely, over a few generations, this mutant variety could have potentially disappeared. It could have been hunted to extinction. One gene could go extinct in a population, leaving all of the other ones still intact. But then the Industrial Revolution happened, and London started burning a lot of coal, and coal creates soot, and soot settles onto everything, including beech trees. So suddenly those nice bright white beech trees looked a lot dimmer, a lot duller, a lot darker. They had a kind of black soot over top of them. And those white peppered moths, they start sticking out like a sore thumb when they land on the beech, but meanwhile, this mutant variety, the dark-winged variety, they get an advantage. Remember that the trait that makes you the most fit is environment dependent. When there was no soot, the white-winged moth was the more fit phenotype. But now that conditions have changed, suddenly the dark-winged moth is the more fit phenotype. So what happened? Well, birds started eating all these white-winged moths. They started eliminating them from the population before they had a chance to breed. Some of them did, but not all of them were able to reach reproductive maturity. Whereas the dark-winged moths had a much higher chance of having offspring. So the next generation's gene pool had more genes for these dark wings. And slowly, the black-winged phenotype became the most common phenotype in that population. So the gene for having this different color came through mutation, but then natural selection increased the population's adaptation to the new conditions that we had. Next cause of evolutionary change we can talk about is genetic drift. Genetic drift is an effect that occurs in small populations, whereby the organisms that happen to mate together just by random chance might have a different distribution of genes than the population as a whole. What do I mean by that? If you take a look at the far left-hand side of this image, we see the first generation that we're going to consider in this example. We have red and white flowers. Now, these flowers follow Mendel's rules of inheritance. So red is the dominant phenotype and white is the recessive phenotype. Now, in this population, we have 10 flowers. Some are red, some are white. Each of them has 2 alleles for this trait. So in total, there's 20 alleles to consider, right? 10 flowers, 2 per flower, we have 20 alleles total. If you count them up, you will find that there are 14 dominant alleles and there are 6 recessive alleles. That means that 70% of the alleles for that gene are dominant and 30% are recessive. These are what we call allele frequencies. How common an allele is in a population is an allele frequency. 70% in generation 1 dominant alleles, 30% in generation 1 recessive alleles. In population genetics problems, we abbreviate the frequency of the dominant allele as a P, and we abbreviate the frequency of the recessive allele as a Q. Don't get mixed up between conventional genetics and population genetics. We're not going to spend nearly as much time doing population genetics problems, so remember capital letters for dominant, lowercase letters for recessive, but this is kind of a special case where we're looking at it a little bit differently. Now let's say, just for instance, that in this particular population of 10 flowers, only 5 of those flowers, about half of the population, successfully reproduces. Well, those flowers that don't reproduce, they're essentially genetic dead ends. As far as the gene pool of the next generation is concerned, they didn't have any contribution. And take a look at those flowers that aren't reproducing. It's mostly flowers with the dominant red phenotype. Not because red is in any way disadvantageous, and it's not holding them back for any reason, it's just random. How are flowers pollinated? Bees come around. And bees see really well in the blue end of the spectrum, but maybe these particular bees don't really care about the difference between red and white flowers, so they just randomly decided to pollinate these particular 5 flowers, the ones that are encircled in this kind of rounded rectangle here. Okay, so only 5 of these flowers breed. So what does the gene pool of the next generation looks like? Well, it certainly isn't going to look exactly the same as the population of the first generation, because half of that population didn't contribute any of their genes to this next generation's gene pool. Not every individual in a population reproduces, so not every individual in a population will have their genes represented in the next generation. So this next generation actually has a 50-50 split between dominant genes and recessive genes. Now let's say for generation 3, only these 2 flowers are pollinated, only these 2 red ones right here. The bees are just doing things entirely by random. They don't have a preference one way or the other. It just so happens that only these 2 red flowers successfully reproduce this particular generation, but there's no recessive alleles in those 2 flowers. So what happens? Recessive allele goes extinct. In the 3rd generation, this right-hand image here, we have no recessive alleles left. This means that we have a 100% dominant allele frequency. We went from 70% to 50% to 100%. In every single generation, the allele frequency changed. That is, by definition, microevolution. That's what we mean when we say the allele frequencies are changing. How common an allele is or is not in a generation might be different from how common it was in previous generations. Now again, genetic drift only affects tiny populations, very small populations. If you have a big population, these types of little random events kind of even out. So you don't see this with big populations. But this does happen a lot on, say, islands where you have very small populations of organisms.